Okay, hi. So I'm Kaitlyn Timmins, and my research project is protecting the planets from microbial contamination, removing and isolating microbes using first contact polymer. So the big question everyone wants to know is there life on other planets? In order to discover this mystery, we need to limit microbial contamination. This can occur both forward and backwards. Forward means that our telescopes would be infected with bacteria and would be traveling to a planet of interest, infecting that planet. Backwards would be that planet would contain its own bacteria that could travel back on our telescopes that could potentially infect Earth. So NASA's Planetary Protection Division is tasked with protecting Earth and other bodies from foreign contamination such as bacteria. Bacteria can survive in extreme conditions such as extreme temperature ranges, high humidity, and the vacuum of space. Bacterial spores are also highly hydrophobic and can adhere tightly to surfaces, which make them extremely hard to remove. So our research project uses First Contact, which is an adhesive polymer demonstrated to clean telescope lenses at an atomic scale. It is in development with NASA to clean the Starshade, which is their newest spacecraft designed to block light of nearby stars to observe orbiting exoplanets. Our research goal was to develop methods that capture and isolate microbes by taking advantage of the unique adhesive properties of the First Contact polymer. So our research design was a two-pronged approach. The first area was the removal assay, which we planted a volume of bacteria on a slide. Typically, we focus on glass for this first project. We let the bacteria dry, and then we added the First Contact, which goes on like a liquid glue and then dries as glue does, so you can peel it off. So once that was dried, we peeled it off and placed it into a tube for the isolation assay to be set aside later. Then we used a soy trypticase broth to resuspend that bacteria, or resuspend that area where that bacteria was originally placed, and it was placed in a test tube where it was heat shocked and then plated overnight to grow. The isolation assay, we used that First Contact from the removal assay, placed that into a test tube, heat shocked, and then added First Contact solvent to dissolve that dried First Contact, and then it was plated to grow. So the results in the reduction assay, this shows the glass. So that big area that you see is the glass that was just bacteria on glass and then resuspended. So you can see that there's obvious growth and that the resuspension works to obtain bacteria from a glass slide. Then the First Contact shows the glass after it was cleaned with First Contact. So after about 8 trials, there was still some area that did have some, but it still shows a 99.1% reduction rate using First Contact. And then with the isolation assay, that chart, the control was just plating the bacteria from a test tube. And then the First Contact was isolating that bacteria from the dried First Contact. And that is about like an 81% isolation rate. So that shows that like we can obtain bacteria from dried First Contact. So in conclusion, First Contact does remove bacterial spores from multiple surfaces. These spores are very difficult though to remove from surfaces. Bacterial spores can also be isolated from the First Contact, and First Contact can also remove DNA, suggesting a use in forensic evidence recovery. So the other conclusions do show that we can reduce the bacteria before a telescope goes into space, and we would be able to isolate bacteria from a telescope if we were to find life on another planet. Our future work would be to expand the reduction and isolation assay to additional surfaces like the NASA recommended surfaces such as titanium and stainless steel. We would like to test NASA provided bacterial and fungal strains isolated from returning spacecrafts, and then use surfactants to reduce the surface tension between First Contact and a surface, as well as perform DNA assays. And then for acknowledgements, I would like to thank Photonic Cleaning Technologies and NASA's Office of Planetary Protection for sponsoring this research, as well as James Hamilton, who invented First Contact, and then Mark Levenstein, who was our advisor on the biology side of this. Thank you.